Hello and welcome. Today I am going to deliver a lecture on regulation of chromatin structure, nucleosome assembly and organization of chromosomes. The main objectives of today's deliberation are: number first, to give a general idea about the chromatin. Number second, to give a precise account of nucleosome assembly. Number third, to give a general and thorough idea of chromosomes. Number fourth, explaining in brief the concept of organization of chromosomes. And finally, explaining regulation of chromatin structure. Now, let me begin by highlighting what chromatin is. Chromatin is a complex of macromolecules found in cells consisting of DNA, protein and RNA. The primary functions of chromatin are: number first, to package DNA into a smaller volume to fit in the cell. Number second, to reinforce the DNA macromolecule to allow mitosis. Number third, to prevent DNA damage. Number fourth, to control gene expression and DNA replication. The primary protein components of chromatin are histones that compact the DNA. Chromatin is only found in eukaryotic cells. Prokaryotic cells have a different organization of their DNA and is localized within the nucleoid region. Chromatin is comprised of histones and DNA. A 147 base pair of DNA chain wrapped around the 8-core histone forms the basic chromatin unit, that is the nucleosome. The function of chromatin is to efficiently package DNA into a small volume to fit into the nucleus of a cell and protect the DNA structure and sequence. Packaging DNA into chromatin allows for mitosis and meiosis, prevents chromosome breakage, and controls gene expression and DNA replication. The structure of chromatin depends on several factors. The overall structure depends on the stage of the cell cycle. During interphase, the chromatin is structurally loose to allow access to RNA and DNA polymerases that transcribe and replicate the DNA. Epigenetic chemical modification of the structural proteins in chromatin also alter the local chromatin structure. In particular, chemical modifications of histone proteins by methylation and acetylation. In general terms, there are three levels of chromatin organization. Number first, DNA wraps around histone proteins forming nucleosomes, the beads on a string structure, that is the euchromatin. Number second, multiple histones wrap into a 30 nanometer fiber consisting of nucleosome arrays in their most compact form, that is the heterochromatin. And number third, higher level DNA packaging of the 30 nanometer fiber into a metaphase chromosome during mitosis and meiosis. Now, let us discuss the nucleosome. A nucleosome is a basic unit of DNA packaging in eukaryotes consisting of a segment of DNA wound in sequence around 8 histone protein cores. This structure is often compared to thread wrapped around a spool. Nucleosomes form the fundamental repeating units of eukaryotic chromatin, which is used to pack the large eukaryotic genome into the nucleus, while still ensuring appropriate access to it. Nucleosomes are folded through a series of successively higher order structures to eventually form a chromosome. This both compacts DNA and creates an added layer of regulatory control which ensures correct gene expression. Besides this, nucleosomes are thought to carry epigenetically inherited information in the form of covalent modifications of their core histones. The nucleosome core particle consists of approximately 147 base pairs of DNA wrapped in 1.67 left-handed superhelical turns around a histone octamer consisting of 2 copies, each of the core histones, that is H2A, H2B, H3 and H4. Core particles are connected by stretches of linker DNA, which can be up to 80 base pairs long. The organization of eukaryotic DNA into chromatin has fundamental implications for our understanding of all cellular processes that use DNA as a substrate. Nucleosome assembly following DNA replication, DNA repair and gene transcription is critical for the maintenance of genomic stability and epigenetic information. Nucleosomes are assembled by replication coupled or replication independent pathways with the aid of histone chaperone proteins. Research studies have provided insight into the mechanisms and the role of histone chaperones in regulating nucleosome assembly. Nucleosome assembly at DNA replication forks requires specific chromatin assembly factors and chaperones. Efficient nucleosome assembly also requires a replication coupled assembly factor, which comprises the anti-silencing function 1 protein and acetylated H3H4 tetramers. An additional histone chaperone is required for H2A, H2B dimers, and this role is thought to be fulfilled by nucleosome assembly protein 1. After newly formed nucleosome core particle arrays are in place, the final assembly of compact chromatin involves the removal of acetyl groups by histone deacetylases. The need for an ordered deposition of histones onto DNA to form nucleosomes arises not only during the replication of DNA in S phase, but also when a cell encounters DNA damage outside S phase or in non-dividing cells undergoing gene expression. Now, coming to the next part of the topic, that is the chromosomes. The idea of chromosomes only appeared in the last quarter of the 19th century. The first scientist to describe clearly the process of mitosis and involvement of the chromatic nuclear figure, that is the chromosomes, was apparently the German zoologist Anton Schneider in 1873. Before then, it was thought that cells and nuclei simply pinched in half to divide. Clear and detailed description of mitotic chromosomes in plants and animals were published by Strasburger in 1875 and by Fleming in 1879-1882 respectively. However, the term chromosome was not introduced until 1888 by Waldeyer, an anatomy professor in Germany. Whatever their function, chromosomes inevitably became popular subjects for study, being conspicuous cellular organelles with considerable aesthetic attraction. However, from the very earliest studies of chromosomes, it became clear that chromosomes were involved in inheritance, so that as early as 1887, Weizmann could put forward his chromosome theory of inheritance. This included the following points. Number first, the nuclear substance controls the form and function of every cell and divides at mitosis to give equal products. Eggs must lose half their nuclear substance in the polar body before fertilization, and this must be replaced exactly by the nuclear substance of the sperm. Because sexual reproduction depends on adding together the egg and sperm nuclei in every generation, there must be a halving of nuclear substance in both male and female germ cells. There are no essential differences between the nuclear substance of eggs and sperm. Sexual reproduction is a means of producing variability between individuals on which natural selection can act. Now coming to the definition of chromosomes. A chromosome is a packaged and organized chromatin, a complex of macromolecules found in cells consisting of DNA and protein. The main information carrying macromolecule is a single piece of coiled double-stranded DNA containing many genes, regulatory elements and other non-coding DNA. The DNA-bound macromolecules are proteins which serve to package the DNA and control its functions. Chromosomes vary widely between different organisms. Chromosomes vary both in number and structure among organisms, and the number of chromosomes is characteristic of every species. Chromosomal recombination during meiosis and subsequent sexual reproduction plays a vital role in genetic diversity. If these structures are manipulated incorrectly through processes known as chromosomal instability and translocation, the cell may undergo mitotic catastrophe and die, or it may unexpectedly evade apoptosis, leading to the progression of cancer. There are normally 2 copies of each chromosome present in every somatic cell. The number of unique chromosomes in such a cell is known as its haploid number, and the total number of chromosomes is its diploid number. The suffix ploid refers to the chromosome sets. The haploid set of the chromosome is also known as the genome. Structurally, eukaryotes possess large linear chromosomes, unlike prokaryotes, which have circular chromosomes. In eukaryotes other than the nucleus, chromosomes are present in mitochondria and chloroplast too. The number of chromosomes in each somatic cell is same for all members of a given species. Now, let us discuss the morphology of chromosomes. First of all, the size. The size of chromosome is normally measured at mitotic metaphase and may be as short as 0.25 micrometer in fungi and birds to as long as 30 micrometer in some plants such as trillium, etc. However, most mitotic chromosomes fall in the range of 3 micrometers in Drosophila to 5 micrometers in man. Next is the shape of chromosomes. The shape of the chromosome changes from phase to phase in the continuous process of cell growth and cell division. During the resting or interphase stage of the cell, the chromosomes occur in the form of thin, coiled elastic and contractile, thread-like stainable structures, the chromatin threads. In the metaphase and the anaphase, the chromosome becomes thick and filamentous. Each chromosome contains a clear zone known as centromere or kinetochore along their length. The centromere divides the chromosome into 2 parts, and each part is called chromosome arm. And last part is the structure of chromosomes. A chromosome at mitotic metaphase consists of 2 symmetrical structures called chromatids. Each chromatid contains a single DNA molecule, and both chromatids are attached to each other by centromere and become separated at the beginning of anaphase. The chromomeres are bead-like accumulations of chromatin material that are sometimes visible along interphase chromosomes. The chromomere bearing chromatin has an appearance of a necklace in which several beads occur on a string. Centromere in a chromosome contains specific DNA sequences with special proteins bound to them, forming a disc-shaped structure called kinetochore. The chromosomes of most organisms contain only one centromere and are known as monocentric chromosomes. Some species have diffused centromeres with microtubules attached along the length of the chromosomes and are termed holocentric chromosomes. Telomere is the chromosomal end which prevents other chromosomal segments to be fused with it. Now, coming to the next part of the topic, that is the forms of chromatin. First is heterochromatin. The chromatin can fold upon itself to compact the nucleosomes, forming a highly condensed structure referred to as heterochromatin. This provides an effective mechanism for storing a large quantity of DNA in a very small volume. However, in order for genes to be transcribed into mRNA molecules, portions of the gene referred to as promoter regions need to interact with particular proteins such as transcription factors that initiate the process of mRNA transcription. In the condensed heterochromatin structure, these promoter regions are largely inaccessible to the transcription factors. Hence, protein production is inhibited. Therefore, to initiate protein production, also referred to as gene expression, the heterochromatin structure needs to be relaxed in a specific and controlled manner so as to expose the promoter regions of particular genes that need to be transcribed at specific times and under specific conditions during the lifetime of a cell. Second is the euchromatin. This more relaxed chromatin structure is referred to as euchromatin. The euchromatin structure exposes the promoter regions of particular genes that need to be transcribed at specific times and under specific conditions during the lifetime of a cell, allowing precisely regulated gene expression and resulting in protein production. The transition from heterochromatin to euchromatin is accomplished by changing the structure of nucleosomes by adding or removing small chemical units, for example, acetyl groups, methyl groups, phosphate groups and ubiquitin groups at specific locations on the histones. These reversible modifications, which result in chromatin relaxation, are performed by selective enzymes within the cell. Proteins participate in shaping chromatin structure, including histone and other chromatin interacting proteins such as transcription factors and the DNA repair machinery. Chromatin remodeling complexes have the ability of changing the chromatin architecture by modulating the interaction between nucleosomes and DNA, which is often achieved by adding post-translational modifications to histones. Heterochromatin is difficult to analyze because of its condensed state and repetitive DNA sequence. Euchromatin poses less challenge as it contains active genes and maintains an open and extended structure. Changes in chromatin structure are at the basis of many regulatory processes. Our understanding of the control of chromatin functions has improved through the identification of components and mechanisms that modify the structural and functional properties of this DNA protein complex. It includes chromatin assembly and remodeling complexes, histone modifications, that is acetylation, methylation, phosphorylation, ubiquitination, structural non-histone proteins, that is HP1 polycomb, which recognize specific histone modifications and contribute to the structural properties of distinct chromatin domains. Histone variants, which localize to specific chromosomal locations. Chromatin structure is influenced by multiple factors such as pH, temperature, nature and concentration of counter ions, post-translational modifications of histones and binding of structural non-histone proteins. RNA is also known to contribute to the regulation of chromatin structure. However, whether RNA itself plays a direct structural role in chromatin remains unclear. Recent studies have revealed that chromatin and nuclear structures are important for the regulation of eukaryotic nuclear function. Nuclear reactions such as transcription and replication are basically repressed in chromatin state. Therefore, changes of chromatin structure mediated by chromatin remodeling factors are crucial. Histones undergo post-translational modifications that alter their interaction with DNA and nuclear proteins. Combinations of modifications are thought to constitute a code, the so-called histone code. Histone modifications act in diverse biological processes such as gene regulation, DNA repair, chromosome condensation and spermatogenesis. Now, let us discuss the functions of histone modifications. A huge catalogue of histone modifications have been described, but a functional understanding of most is still lacking. Collectively, it is thought that histone modifications may underlie a histone code whereby combinations of histone modifications have specific meanings. Now coming to the chromatin remodeling. Chromatin remodeling is the dynamic modification of chromatin architecture to allow access of condensed genomic DNA to the regulatory transcription machinery proteins and thereby control gene expression. Such remodeling is principally carried out by number first, covalent histone modifications by specific enzymes, that is histone acetyltransferases, deacetylases, methyltransferases and kinases. Number second, ATP dependent chromatin remodeling complexes which either move, eject or restructure nucleosomes. Besides actively regulating gene expression, dynamic remodeling of chromatin imparts an epigenetic regulatory role in several key biological processes. For example, DNA replication and repair, apoptosis, chromosome segregation as well as development and pluripotency. Aberrations in chromatin remodeling proteins are found to be associated with human diseases including cancer. Targeting chromatin remodeling pathways is currently evolving as a major therapeutic strategy in the treatment of several cancers. Now, let us try to learn about covalent histone modifying complexes. Specific protein complexes known as histone modifying complexes catalyze addition or removal of various chemical elements on histones. These enzymatic modifications include acetylation, methylation, phosphorylation and ubiquitination and primarily occur at N-terminal histone tails. Such modifications affect the binding affinity between histones and DNA and thus loosening and tightening the condensed DNA wrapped around histones. For example, methylation of specific lysine residues in H3 and H4 causes further condensation of DNA around histones and thereby preventing binding of transcription factors to the DNA leading to gene repression. On contrary, histone acetylation relaxes chromatin condensation and exposes DNA for TF binding leading to increased gene expression. Well characterized modifications to histones include number first is the methylation. Both lysine and arginine residues are known to be methylated. Methylated lysines are the best understood marks of the histone code as specific methylated lysine match well with gene expression states. Number second is acetylation by histone acetyltransferases, deacetylation by histone deacetylases. Acetylation tends to define the openness of chromatin as acetylated histones cannot pack as well together as deacetylated histones. Third is phosphorylation. Like histone acetylation, the phosphorylation of histones is highly dynamic. It takes place on serines, threonines and tyrosines, predominantly but not exclusively in the N-terminal histone tails. The levels of the modification are controlled by kinases and phosphatases that add and remove the modification respectively. Fourth is ubiquitination. All of the previously described histone modifications result in relatively small molecular changes to amino acid side chains. In contrast, ubiquitination results in a much larger covalent modification. Ubiquitin itself is a 76 amino acid polypeptide that is attached to histone lysines via the sequential action of 3 enzymes, that is E1 activating, E2 conjugating and E3 ligating enzymes. The enzyme complexes determine both substrate specificity, that is which lysine is targeted, as well as the degree of ubiquitination, that is either mono or polyubiquitinated. Last one is the histone code hypothesis. The critical concept of the histone code hypothesis is that the histone modifications serve to recruit other proteins by specific recognition of the modified histone via protein domains specialized for such purposes, rather than through simply stabilizing or destabilizing the interaction between histone and the underlying DNA. These recruited proteins then acted to alter chromatin structure actively or to promote transcription. After learning about covalent histone modifying complexes, now let us try to learn about ATP dependent chromatin remodeling. ATP dependent chromatin remodeling complexes regulate gene expression by either moving, ejecting or restructuring nucleosomes. These protein complexes have a common ATPase domain, and energy from the hydrolysis of ATP allows these remodeling complexes to slide, twist or loop nucleosomes along the DNA, expel histones from DNA or facilitate exchange of histone variants, and thus creating nucleosome free regions of DNA for gene activation. Also, several remodelers have DNA translocation activity to carry out specific remodeling tasks. At last, we will learn about the significance of chromatin remodeling. Number first is in normal biological processes. Chromatin remodeling plays a central role in the regulation of gene expression by providing transcription machinery with dynamic access to an otherwise tightly packaged genome. Further, nucleosome movement by chromatin remodelers is essential to several important biological processes, including chromosome assembly and segregation, DNA replication and repair, embryonic development and pluripotency, and cell cycle progression. Deregulation of chromatin remodeling causes loss of transcriptional regulation at these critical checkpoints required for proper cellular function, and thus causes various disease syndromes including cancer. Number second is in cancer. Chromatin remodeling provides the fine tuning at crucial cell growth and division steps like cell cycle progression, DNA repair and chromosome segregation, and therefore exerts tumor suppressor function. Mutations in such chromatin remodelers and deregulated covalent histone modifications potentially favor self-sufficiency in cell growth and escape from growth regulatory cell signals that are the two important hallmarks of cancer. Number third is in genomics. Rapid advance in cancer genomics and sequencing methods are providing more insight into the role of chromatin remodeling in transcriptional regulation and role in cancer. And the final is in therapeutic intervention. Epigenetic instability caused by deregulation in chromatin remodeling is studied in several cancers, including breast cancer, colorectal cancer, pancreatic cancer, etc. Such instability largely causes widespread silencing of genes with primary impact on tumor suppressor genes. Hence, strategies are now being tried to overcome epigenetic silencing with synergistic combination of HDAC inhibitors or HDI and DNA demethylating agents. That's all we have for you in today's lecture. Hope you have understood it well. Thank you very much and goodbye.